Mineral Nutrition Part 2. In isolated cells, tissues or organs, much of the studies on mechanism of absorption of elements by plants has been carried out. These studies reveal that into two main phases the absorption process can be demarcated. In the first place, an initial rapid uptake of ions into the free space or outer space of cells, the apoplast, is passive. In the second phase of uptake, slowly the ions are taken into the inner space, the symplast of the cells. Usually into the apoplast, the passive movement of ions occurs through ion channels, the transmembrane proteins that function as selective pores. The entry or exit of ions to and from the symplast, on the other hand, requires the expenditure of metabolic energy, which is an active process. Usually the movement of ions is called flux. Influx is the inward movement into the cells and the outward movement is efflux. Translocation of solutes. Along with the ascending stream of water, mineral salts are translocated through xylem, which is pulled up by transpirational pull through the plant. The presence of mineral salts are shown by the analysis of xylem sap. The radioisotopes of mineral elements also provide evidence to support the view that they are transported through the xylem. Soil as reservoir of essential elements. Due to withering and breakdown of rocks, majority of the nutrients that are essential for the growth and development of plants became available to the roots. With dissolved ions and inorganic salts, these processes enrich the soil. Since the minerals are derived from the rock, their role in plant nutrition is referred to as mineral nutrition. A wide variety of substances are present in the soil. Along with minerals, soil also holds water, supplies air to the roots, harbors nitrogen-fixing bacteria, other microbes, and acts as a matrix that stabilizes the plant. Since the crop yield is affected by the deficiency of essential minerals, there is often a need for supplying them through fertilizers. Components of fertilizers, both macronutrients and micronutrients, are applied as per need. Metabolism of nitrogen, nitrogen cycle. In living organisms, the most prevalent element is nitrogen apart from carbon, hydrogen, and oxygen. Amino acids, proteins, hormones, chlorophylls, and in many of the vitamins, nitrogen is present. For the limited nitrogen that is available in soil, plants compete with microbes. For both natural and agricultural ecosystems, nitrogen is thus a limiting nutrient. As two nitrogen atoms, nitrogen exists and is joined by a very strong triple covalent bond. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. In nature, to convert nitrogen to nitrogen oxides, enough energy is provided by lightning and ultraviolet radiation. Also, the sources of atmospheric nitrogen oxides are industrial combustion, forest fires, automobile exhaust, and power generating stations. The process of decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. A small amount of ammonia volatilizes and re-enters the atmosphere, but most of it is converted by soil bacteria into nitrate in the following steps. By the bacteria Nitrosomonas and or Nitrococcus, ammonia is first oxidized to nitrite. With the help of the bacterium Nitrobacter, the nitrite is further oxidized to nitrate. These steps are called nitrification, and these nitrifying bacteria are chemoautotrophs. By plants, the nitrate thus formed is absorbed and is transported to the leaves. It is reduced to form ammonia in leaves and finally forms the amine group of amino acids. By the process of denitrification, the nitrate in the soil is also reduced to nitrogen. The bacteria Pseudomonas and Theobacillus carry out the process of denitrification, biological nitrogen fixation, in the form of nitrogen which is available abundantly in the air. Very few living organisms can utilize the nitrogen. Only certain prokaryotic species are capable of fixing nitrogen. By living organisms, reduction of nitrogen to ammonia is called biological nitrogen fixation. Exclusively in prokaryotes, the enzyme nitrogenase, which is capable of reducing nitrogen, is present. Such microbes are called nitrogen fixers. The nitrogen fixing microbes could be free living or symbiotic. Examples of nitrogen-fixing aerobic microbes that are free-living are Azotobacter and Bijrinkia, while anaerobic microbe is Rhodospirillum and Bacillus is free-living. Also free-living nitrogen fixers are a number of cyanobacteria such as Anabina and Nostoc. Symbiotic biological nitrogen fixation. There are several types of symbiotic biological nitrogen fixing associations. The legume bacteria relationship is the most prominent among them. Rod-shaped Rhizobium species has relationship with the roots of several legumes such as sweet pea, clover beans, alfalfa, sweet clover, garden peas, lentils, broad bean, etc. On roots, the most common association is as nodules. Small outgrowths on the roots are the nodules. On the roots of non-leguminous plants, example Alnus, the microbe Frankia produces nitrogen-fixing nodules. In the soil, both Rhizobium and Frankia are free-living, but they can fix atmospheric nitrogen as symbionts. Just before flowering, if any one plant of a common pulse is uprooted, the near spherical outgrowths on the roots can be seen. These are nodules. You will notice that the central portion is red or pink if you cut through them. What makes the nodules pink? Due to the presence of leguminous hemoglobin or leg hemoglobin, the nodules are pink. Nodule formation. In between rhizobium and roots of the host plant, the nodule formation involves a sequence of multiple interactions. In the nodule formation, principal stages are summarized as follows. In the surroundings of roots, rhizobia multiply and colonize and get attached to epidermal and root hair cells. The bacteria invade the root hair as the root hair curls. Into the cortex of the root, an infection thread is produced carrying the bacteria, and in the cortex of the root they initiate the nodule formation. In the cells, the bacteria are released from the thread, which leads to the differentiation of specialized nitrogen-fixing cells. When the nodule is formed, a direct vascular connection is established for exchange of nutrients with the host. All the necessary biochemical components are present in the nodule, such as the leg hemoglobin and enzyme nitrogenase. The molybdenum iron protein enzyme nitrogenase catalyzes the conversion of atmospheric nitrogen to ammonia, which is the first stable product of nitrogen fixation. The reaction is as follows. To the molecular oxygen, the enzyme nitrogenase is highly sensitive and it requires anaerobic conditions. To ensure that the enzyme is protected from oxygen, the nodules have adaptations. The nodule contains an oxygen scavenger called leg hemoglobin to protect these enzymes. Under free living conditions where nitrogenase is not operational, it is interesting to note that these microbes live as aerobes but they become anaerobic, thus protecting the nitrogenase enzyme during nitrogen fixing events. In the above reaction, you must have noticed that the ammonia synthesized by nitrogenase requires a very high input of energy. From the respiration of the host cells, the energy required is obtained. Fate of ammonia. The ammonia is protonated to form ammonium ion at a physiological pH. Most of the plants can assimilate ammonium ions as well as nitrate. The former is quite toxic to the plants and hence cannot accumulate in them. Let us see how the ammonium ion is used to synthesize amino acids in plants. There are two main ways in which this can take place. 1. Reductive amination. 2. Transamination. 1. Reductive amination. Ammonia reacts with alpha-ketoglutaric acid and forms glutamic acid in these processes as indicated in the equation given below. 2. Transamination. This involves the transfer of amino group from one amino acid to the keto acid of a keto group. The main amino acid, the glutamic acid from which the transfer of NH to the amino group takes place, and through transamination other amino acids are formed. All such reactions are catalyzed by the enzyme transaminase. For example, the two most important amides found in plants, namely asparagine and glutamine, are the structural part of proteins. From two amino acids, namely aspartic acid and glutamic acid, they are formed by addition of another amino group to each. By another amino radical, the hydroxyl part of the acid is replaced. Since the amino acids do not contain more nitrogen than amides via xylem vessels, they are transported to other parts of the plant. The nodules of some plants export the fixed nitrogen as ureates along with the transpiration stream. A particularly high nitrogen to carbon ratio is present in these compounds. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please click the description box down below.